Welcome to this platform. So our main focus here in this video, we are going to look at enzymes. We are going to focus mainly on characteristics of enzymes and also the industrial applications of enzymes. But before then, we'll have to look at some other concept related to enzymes. And therefore, the first question that we are answering is, what is an enzyme? What is an enzyme? So an enzyme is a biological catalyst. Is that simply a biological catalyst? That is how we define an enzyme. Now, there's a word that we need to understand here, which is a catalyst. If an enzyme is a biological catalyst, then what does it do in the body of living organisms? So a catalyst is just a chemical substance that speeds up a rate of chemical reactions without it being changed. Okay, that is how we define a catalyst. That's a chemical substance that speeds up rate of chemical reaction without itself being changed at the end of the reaction. So this means that a biological catalyst, or an enzyme rather, speed up rate of chemical reaction that occur in the cells. All right, now depending with where they work, enzymes can be classified into two. Okay, depending where they perform their functions. You know, enzymes are produced in the cells. Now, there are those enzymes that work within the cells where they are produced, and these are called intracellular enzymes. So enzymes that work within the cells they are produced are called intracellular enzymes. So intracellular enzymes, in other words, we can say enzymes that work within the cell they are produced. Then, of course, they have also those that are transported to where the chemical reaction is taking place. They are produced in a cell and, of course, moved to the parts where the reaction is taking place. And these are called extracellular enzymes. In short, extracellular enzymes are enzymes that work outside the cell. So two types of enzymes that you need to take note, that is intracellular and extracellular, based on where they work. Okay. So now, it is very important also, as we've talked of the enzyme, it's very important we look at the general parts of an enzyme. Now, this drawing that you are able to see here, I'm not saying that every time the enzyme is going to be presented as such, but this is just a general. Okay, so here is the part of an enzyme. And what do we call this part? Let me start with this. Let me say this is X and this is Y. So two parts of the enzyme here. X is called allosteric site. Okay, then of course we have Y pointing on these two spaces here, and these are what, or this is what, the active site. Okay, the active site. Now as we go on, the enzyme acts on the substrate. So we have another term that we need to understand, which is the substrate, of which is just a substance on which an enzyme acts on. As we go on, we are going to look at the theory that we are going to look at for the sake of learning. Okay. So now, aside from looking at this part, that is the parts of an enzyme. So please, you should take note of these parts. As we go on in our discussion, we are going to refer to these parts most often. Now, the other thing that we can look at is the general enzyme catalyzed reaction, how the enzyme work out, or how it speed up the reaction, or how it helps in speeding up the reaction. So that is what we call the general catalyzed reaction. Okay. So now what happens normally is that the enzyme and the substrate bind to the enzyme, then they form what we call enzyme substrate complex, and now that is where now it's split into enzymes and the product. Only when the enzyme substrate complex is formed, that is when the products are formed. So in other words, the enzyme, for it to break down the substrate bind to the active site of an enzyme, then it forms the enzyme substrate complex, which later breaks into the enzyme and the product. As you know, that an enzyme is a catalyst, and therefore at the end of the reaction it remains as an enzyme. Okay, ready to take on some more substrate. All right, so that is something that you should understand. Now, about characteristics of an enzyme, that is one of the parts that we are going to look at, or that is one of our main objectives that we are going to look at. So what are the characteristics of enzymes? Okay, so these are general, whether these are characteristics that you are going even to come across, even if you do that is biology, that is biochemistry at an advanced level, you're still going to find these characteristics as the same. So characteristics of enzyme, we have one, they are protein in nature. So all enzymes are protein in nature. And as we look at the factors that affect enzyme activity, we are going to see and of course understand this very well. So they are protein in nature. So if you're asked of the characteristics of enzymes, if you say they are protein in nature, then you're 100% all right. Then they catalyze both forward and reverse reaction, which means the enzyme reaction are reversible, as you can see from the half arrow here. So we have the arrow, half of it this side, half of it going this side. This means that the reactions is reversible. So enzyme substrate to enzyme substrate complex, and from enzyme substrate complex to enzyme and product, you can also move from here going this side. So the reaction is reversible. So they both are catalyzed, that is forward and reverse reaction. Their activities is affected by temperature, pH, substrate concentration, enzyme concentration, inhibitors, and cofactors. So these are the factors that we are going to discuss as the factors that affect enzyme activity, and we are going to see how they do that. Okay, so that is one of the characteristic. Then of course we can look at the other one, that is they are specific. So the other characteristic is that they are specific. Now this is where we look at the theory called lock and key theory mechanism. So in this lock and key theory mechanism, it looks at the active site of an enzyme being the lock, the enzyme being the lock, and of course the substrate being the key. As you know, if you were to open a certain lock, only specific keys do open there, because only it has, each and every lock has specific keys. Okay, so that is how you look at it. So enzymes, in other words, they act on specific substrate, or they have a narrow range of substrate they act on. They cannot act on every kind of substrate. They have specific substrate. For example, they are enzymes that break down proteins, so specifically for proteins. Then carbohydrates, specifically for carbohydrates. So that is what it means, the enzymes are specific. So that is one of the characteristics that you need to know. And in terms of specificity, we look at the lock and the key model or mechanism for a meantime. But as you go on, you are going to find other theories that you're going to come across, that is for this part. All right, so having looked at the characteristics of enzymes, the other thing that we can look at are the factors that affect enzyme activities. Now, from where we're coming from, we are saying enzymes activity, or they are affected by temperature, pH, substrate, enzyme concentration, inhibitors, and cofactors. So under factors that affect enzyme activity, these are the ones that we are going to discuss. So factors that affect enzyme activity, one, we have temperature. Now remember what we said, enzymes are protein in nature. All right, now let us look at how temperature affects enzyme activity. So it happens that the increase in temperature increases enzyme activity up to a certain point called the optimum temperature. So increase in temperature increase enzyme activity until it reaches and up to the certain temperature called the optimum temperature. Now the optimum temperature, for you to know, is just the temperature at which enzyme has maximum activity, or it works best. That is what we call the optimum temperature. And of course we are going to look at the graphical presentation very soon. So now, why does why does temperature increase enzyme activity? We look at it in detail. So the reason behind is that because the increase in temperature results into the increase in kinetic energy of both enzymes and the substrate, leading to increased interaction between the enzyme and the substrate, and this lead to the formation of what we call enzyme substrate complex. Remember what we said from the start, that only if the enzyme substrate complex is formed, that is when now the products can be formed. Okay, so that is how it affects the enzyme activity. So in other words, temperature increases enzyme activity, because affecting, we are looking at, does it lower or increase? So that is mainly that is what we need to understand here. Then the other one, okay, so maybe just to add to that, we are saying activity reduces beyond the optimum temperature because the enzyme get denatured. So we have a new word here, which is denatured. So which means beyond the part which is called the optimum temperature, the enzyme get denatured. Now, enzyme denaturation can be permanent or temporal. Temporal, which means it can be reversed, but can be permanent also, which means the enzyme is completely dead. Okay, now we are going to look at exactly what happened. Why does the activity reduces when the enzyme get denatured? So in terms of graphical representation, this is how it looks. So rate of enzyme catalyzed reaction, as you can see from this side, then we have temperature this side. So what this graph shows is that from here, which is 0, going up here, the rate of chemical reaction or enzyme catalyzed reaction increases. Then going this side, also we are increasing temperature. So as you increase the temperature, also rate of enzyme activity increases until the point X there. Here, that is where you find the optimum temperature. So at the optimum temperature, that is where the enzyme has maximum activity. But when it goes beyond, which means the temperature has gone beyond the optimum temperature, if you have an enzyme like this one, let's say it was like this, the active site was like this, then when it go beyond the optimum temperature, the active site is disrupted. Maybe it becomes like this. So which means the substrate that fitted here, it cannot fit here. As a result, no reaction occurs. Remember, only when the enzyme substrate complex forms, that is when the product can form. And in this case, if the substrate does not bind to this or does not fit well to the active site, then no reaction occurs. And that is the reason why the reaction or the activity reduces. So this is about enzyme, this is about temperature affecting, or this is how temperature affect activity of an enzyme. The other factor that we can look at is pH. Okay, now pH, written as this, we can say it's potential hydrogen. Now this is just a measure of how acidity or alkalinity the substance is. Okay, that is pH. So pH has to do with substance that is either alkaline, acidic, or neutral. Okay, but how does it affect enzyme activity? How does it affect enzyme activity? So to start with, we need to look at the pH scale. So the pH scale, we can start from here, that is 1 all the way up to 14. So from 7 going up, that is alkaline. So as you go up to 14, that is the more alkaline the solution or substance become. Then as you come down 7 below, it become more acidic, which means the pH of 2 is more acidic compared to the pH of 6. So going this side, as you approach 1 here, the acidity increases. Now 7 is neutral. Now how does pH affect the enzyme activity? Now different enzymes work well in certain pH. For example, the enzymes that work in the stomach, such as pepsin, they work well in acidic environment. As you know, the stomach is acidic. Well, if that information is new, we are going to look at digestion. That is where we get to discuss that. But of course, just to make you know that the stomach is acidic. But as you go along the, that is the intestine, in the duodenum, the pH there is alkaline. So the enzyme that survive in the stomach, it cannot, or that work well in the stomach, cannot work in the alkaline condition, that is in the duodenum or in the mouth. And also the one that work well in the mouth cannot perform its function in the stomach. So which means they have a certain pH, they work well, and that is what we call optimum pH. So pH value at which the enzyme works well is called the optimum pH. Let us look at the graphical presentation. So this is how it looks in terms of a graph. Equally, we have from 0 here going up, rate of enzyme activity increases, then going this side we're increasing the pH. So as you start from here, as the pH increases, rate of enzyme activity also increases, but it reaches at the point where the enzyme has maximum activity, and that is what we call optimum pH. In an exam, you might find graphs like this. Then you're asked maybe give the optimum pH. You have numbers, let's say 2, that is let's say 14, let's say 0, this is the 2. I'm just giving an example. The optimum pH, you just pick 7 here. So the top part here, you just put a line, then you pick the number there. So that is basically what you might encounter. So that is the optimum pH. Now, beyond the optimum pH, as you can see, starting from this point, going this side, the activity is what? Is decreasing. And that is because it has gone beyond optimum pH. Beyond optimum pH, it causes an enzyme to lose its active site, the shape of its active site. As a result, enzymes cannot bind or fit, substrate cannot fit properly, and therefore no reaction or reaction reduces. At this point, the enzyme is completely denatured. So from here, denaturation has started, but as it reaches or touches this part, there's completely denaturation. Some questions that we expect is that at what temperature or at what pH was the enzyme completely denatured? So it is completely denatured at a point where it touches this line. I guess you've gotten that. All right, so that is all about pH. The other one that we can discuss is enzyme concentration. Now, enzyme concentration, how does it affect the activity of the enzyme? In this case, you are dealing with enzymes and also the substrate. So you hold the enzyme constant. Okay, the enzyme concentration. Is it the enzyme concentration? The rate of enzyme activity reaction measures with the increase in concentration of enzyme remain the constant where. Okay, let us look at it. So the rate of enzyme catalyzed reaction increases with increase in the concentration of what? Enzyme, and remain constant when there is no more free substrate molecules for the enzyme to act. So in this case, you are increasing the enzyme, then you hold the concentration of the substrate constant. So as you increase the concentration of the enzymes, you are going to find that you have more enzymes but no substrate to act on. So at that particular point where you have no substrate in the solution, the reaction becomes constant. So as you can see, rate of enzyme catalyzed reaction, then of course enzyme concentration. So as you increase the enzyme concentration, the rate of reaction increases, but you reach at the point where all the enzymes are, all the substrate have been catalyzed. At that point we say, so this point we can say all the substrate molecule are used up. So at this point, all the substrate molecules are used up. So at that particular point. All right. The other one that we can look at is substrate concentration. So for substrate concentration, the rate of an enzyme catalyzed reaction increases with increase in the what? Concentration of a substrate, until the level where all the enzyme active sites are occupied by the substrate molecules. So this time around, you are holding the enzyme constant, and now you are increasing the concentration of the what? The substrate. So equally, this one occurs more like the same as enzyme concentration. So as we increase as we increase the concentration of a substrate, you find that the reaction continue increasing until at the point where all enzymes active site are occupied. Okay, so that is how it looks like, or that is how it looks graphically. Then the other factor that we can look at are inhibitors. So inhibitors, or inhibitor, is just any chemical substance that slows down completely or stops enzyme activity. That is what we call inhibitor. Now, since an inhibitor is any substance that slows down or completely stops enzyme activity, we can classify them into two. So we have what we call competitive inhibitors and non-competitive inhibitors. Now, if this is an enzyme, and that is the active site, competitive inhibitors compete for an active site with the substrate. So when the inhibitor binds first, it blocks the substrate from binding. As a result, the reaction is slow, because only when the substrate bind there, then that is when the reaction occurs. But for non-competitive, it binds on the allosteric site and deform the shape of the active site, hence making a substrate not fit on the active site. Okay, so here we can say competitive inhibitors bind to the active site and block the substrate from binding there. I think that is a simplified way. Then, of course, non-competitive, we are saying it binds to the allosteric site of an enzyme and cause the shape of the active site to change so that the substrate does not fit anymore. So that is how the non-competitive inhibitors works. The other factor that we can look at are cofactors. Now, cofactors, they act more like helpers. They enhance the enzyme activity. Okay, they increase the enzyme power to break down substrate. So in other words, they work alongside with enzymes. So it's any non-protein substance whose presence makes an enzyme active. Example, we have vitamins. All those are cofactors. All right, so these are the factors that we can look at in terms of the factors that affect enzyme activity, and of course we've discussed how that occurs. Now, what is the question that you might expect in an exam? That is on essay question to start with. You might be given a question to explain the factors that affect enzyme activity, and if that question comes, you should be able to explain everything you should be able to write everything that I've explained. Okay, then of course we can look at the naming of enzymes. How are the enzymes named? Okay, because as we go along, we are going to find different types of enzymes, and you might be wondering, how are these enzymes named? So the naming system of enzyme take into account the substrate on which the enzyme act on. So for example, if you have the substrate, which is the maltose, okay, maltose. So maltose, the enzyme that act on maltose is called maltase. Okay, maltase. So naming of enzymes, the first part comes from the substrate, and the ending part, which is the ASE. Okay, that is that makes the enzyme. So maltose, maltase. So 3 parts or 3 letters at the end, you add this one. The other one that we can look at on the examples is lactose. So lactose, you can guess the enzyme will be lactase. Then also we have sucrose. Sucrose, the end or the name of the enzyme will be sucrase. Although not all enzymes are named this way, but that is how most of the enzymes are named. Okay, so it's very important to understand this part. All right, so we are going to end by looking at industrial application of enzymes. What are the application of enzymes at industrial level or in industries? So one of the application of enzymes in industry is making of biological detergents. So making of biological detergents is one of the application. So enzymes are included in the biological detergents so that they can hydrolyze the stains of biological origin. Now, you are aware that we use much of, the detergents, one of the use is washing clothes. And of course, the stains that we remove from the clothes might be protein stains, fat stains. These biological detergents, for example, if you stay in Zambia, we have Boom, Bullet, and all sort of detergents that we have. So those are biological, or they contain enzymes. They are called biological detergents. The other use of the other industrial use of enzyme, it is used in what? Brewing and baking. So in baking and brewing, make use of enzyme found in yeast. You know that in brewing and baking they make use of yeast. Yeast contain an enzyme called enzymes, and therefore it makes it possible for brewing and baking. So the other part here, you are going to this part you are going to research on other application. Let me just give you the application so that you get to research on how they are used. So I can add the third one, which is making. So the third one, we can say making of sweeteners. Making of sweeteners for foods and drinks. Okay, making sweeteners for food and drinks. Then it is used in the daily industry. So daily industry, what is it used for? So please make sure that you go through these parts. So we've come to the end of the lesson, please. And if you are interested to join us for online lessons, you can contact the number 0976 402563. And if you know someone who is writing also, you can refer them to this platform. Otherwise, we can end here and see you in the next one.